Barbara Melchert, and I'm from the Department of Molecular Pharmacology of the University of Groningen. Today's date is March 20th, and this is a recorded class about immunity and immune cells for the course Pathology and Immunology for the first year pharmacy students. I originally gave this class on March 3rd, but I re-recorded it for a complete set of online lectures. So, more about the immune system. What do we have the immune system for? Well, basically for the protection of self through law and order. The immune system is very much like a police state, basically. And when we are threatened, then they go into lockdown and start protecting self by raising barriers, by having lots of police around, and this can be seen in the case of this new coronavirus. I talked about that during the lecture a lot, but I've now made separate lectures on coronavirus, so you can have more background on that in the other recorded lectures. But the new coronavirus is similar to outbreaks we've had before with the severe acute respiratory syndrome, or SARS, and the Middle East respiratory syndrome, MERS. And this is a variant that is very much like the first SARS virus in 2002-2003, with a similar mortality rate and spread. Back in 2002 it was very well contained, but in 2019 this went wrong, probably because it was it developed in a very different time. It developed in a very busy metropole in China during a time when many people travel, the Chinese New Year, which really augmented the spread of the disease. So, but this is a new virus. There are no vaccinations. Nobody in the world has seen it before. So how does your immune system deal with such a new threat? Well, first of all, we are protecting ourselves by barriers. So we have epithelial barriers on all the surfaces that are exposed to the outside. So the skin, the gut, the lung, they all have very tightly packed epithelial barriers to keep threats out. And these epithelial barriers are also have cellular alarm systems that can tell the cells what is happening outside, that they need to protect themselves, that they need to ramp up antiviral responses. And these epithelial barriers have tissue-resident immune cells. It's sort of like a neighborhood watch. They patrol these tissues that are exposed to the outside world and help with the first response when there is a breach of the epithelial barriers. They try to solve the problem. If they cannot solve the problem, they will call in the help from the bone marrow. So that's neutrophils and monocytes that are circulating in the blood, that they can call in for extra help and try to fight, in this case, an infection. If that doesn't work, then they can call in the help from very specialized immune cells called lymphocytes that they can ask for help. And what do these lymphocytes do? Well, we have three types. We have lymphocytes that help other cells to fight. We have and they're called T helper cells, or CD4 positive T cells. We have T lymphocytes that can actually kill other cells themselves. and we call them cytotoxic T lymphocytes, or CD8 positive T cells. And we have B lymphocytes. Those are the cells that produce antibodies and can help out with reducing the spread of a virus in the body. So where do all these immune cells come from? Well, all leukocytes, and that's what we call white blood cells, develop from either stem cells in bone marrow or from progenitors in the fetal yolk sac or the fetal liver. And a stem cell can develop into a progenitor cell that can then differentiate into many other types of cells. So what do we have circulating in our blood? Well, we have monocytes, eosinophils, basophils, lymphocytes, and neutrophils, and they all have different functions, and they are important in different parts of the immune response that we'll talk about in later classes. So this picture depicts the most important characteristics of these cells, and what you can see is that some cells have granules inside, like eosinophils and basophils and neutrophils. Some cells are a little bit bigger, like monocytes, and some cells basically are a nucleus with not much else. So what is very characteristic for monocytes is that they're slightly bigger, they have more cytoplasm, and their nucleus is heart-shaped. And then we have three types of cells that we also call granulocytes, because they have granules inside. Those are eosinophils, basophils, and neutrophils. And they are called that way because these granules respond differently to stainings that we can do to visualize these cells. So eosinophils have granules that like acid, so that's why they're called eosinophil. Eosino means acid and phil means to love. And these granules can be stained with pink, with a acidic stain, while the basophil has granules that they like a more alkaline stain, so they stain blue with an alkaline stain, and baso means alkaline. And neutrophils have granules that do not stain with an acidic or an alkaline stain. They are neutral, and that's why they're called neutrophils. So all these cells have granules. Eosinophils are pink, basophils are blue, and these are sort of non-colored. But what is also characteristic is that they have a segmented nucleus. So the nucleus is not one big blob, but it's divided into parts, and particularly the neutrophils has many lobules. The eosinophil usually has two lobules and sort of looks like a headphone or sunglasses, and the basophil sort of in between these two. That leaves us with lymphocytes. Lymphocytes are usually a bit smaller, and they don't have a lot of cytoplasm. They have a very big nucleus. And if you then look in blood to see what cells are circulating, then most of the white blood cells that are circulating are neutrophils, then lymphocytes, then monocytes, then eosinophils. But these can increase a lot in people who suffer from allergies or asthma. Then this particular cell type is increasingly present. And then the cells that are fewest in blood are basophils. And these are actual stainings of the different cells. The basophil with the blue granules and the segmented nucleus here, then the eosinophil with the headphone nucleus and the pink granules, lymphocyte with very little cytoplasm but the big nucleus, then the monocytes with a lot of cytoplasm and the heart-shaped nucleus, and the neutrophil that doesn't have, from which the granules are not really stained, but it has a very segmented nucleus. So how do these immune cells develop? Well, they develop from a pluripotent hematopoietic stem cell in bone marrow, and this stem cell can choose to become a lymphocyte progenitor or a myeloid progenitor. If it becomes a lymphocyte progenitor, then it can turn into a T cell, into a B cell, or a natural killer cell. So within bone marrow, the lymphocytes can develop into a B cell. But to become a T cell, this lymphocyte progenitor has to move to the thymus, and in the thymus it can develop into a proper T lymphocyte. So these cells all circulate, but they can also decide to go into tissues. And for the B cell, when the B cell becomes activated and starts producing antibodies, it will move into the tissues and become a plasma cell. So the myeloid stem cell can actually become a lot of cells. It can become a red blood cell, an erythrocyte, or platelets, but also the granulocytes, eosinophils, basophils, and neutrophils, or a monocyte, and even a mast cell or a dendritic cell. And these do not have circulating siblings. These go directly to the tissue to develop into a mast cell or a dendritic cell. In the tissues we also have macrophages that got their name because they're big eaters. Macro, big, phage, eat. These can either develop from monocytes that go into tissues, or they can be tissue resident and self-renew in the tissues. So what is the difference exactly between a monocyte and a macrophage? A monocyte and a macrophage are in some ways very similar and in some ways are not. The monocyte is a circulating cell. It can be a precursor of a tissue macrophage, and it's recruited to sites of inflammation. So it's circulating in blood, and when a tissue calls out for help, it will go to the tissue to help out with an infection, for instance. A macrophage is very important for phagocytosis, bacterial killing, and antigen presentation. We'll talk about that later more. But they're tissue resident. They don't circulate. They're present in tissues, and they can either become tissue resident because a monocyte goes into the tissue and becomes a macrophage, but many of the tissues are seeded during fetal development with progenitors from the fetus. So either the yolk sac around the fetus can supply progenitors that go to the tissues of the fetus and become tissue resident macrophages, or the fetal liver can provide progenitors that go to the tissues and become tissue resident macrophages. And these cells then sort of have a stem cell function in those tissues and self-renew when it's necessary. So, for instance, the microglia, the skin, the lung, and the liver, they have macrophages that have developed from these fetal progenitors. And when they need more of these macrophages, the tissue-resident macrophages themselves will proliferate, self-renew, and make sure that there are enough macrophages in the tissue. Of course, they can get help from monocytes that go into the tissue and become a macrophage, but the main renewal in these tissues is from self-renewal of the macrophages that are present. And some tissues, they initially have these fetal progenitors that become macrophages, but then when the fetus is born, the monocytes take over and make sure that there are enough tissue-resident macrophages, and that's particularly important for the intestines where there's a lot of turnover of macrophages, and they're all derived from monocytes. So we have two types of progenitors and two types of immune systems. We have the innate immune system and the adaptive immune system. So most of the myeloid cells belong to the innate immune system, and most of the lymphoid cells belong to the adaptive immune system. And there are some cells that are in between that are derived from lymphoid progenitors but actually have an innate immune function. And I've made a list here. Dendritic cells, macrophages, neutrophils, eosinophils, basophils, and mast cells are all part of the innate immune system and myeloid cells. B and T cells are part of the adaptive immune cells and part of and from lymphoid progenitors. But these cells are derived from lymphoid progenitors, but actually have innate immune function. During immunopharmacology in the second year, we'll talk more about these cells. For this course, these are the most important. So together, all these cells constitute the two immune systems that work together to fight off threats, for instance, an infection. So the innate immune system is the system that is fast and is the first response against an infection. And then when they cannot clear an infection, then they call in the help from the adaptive immune system, which takes a longer time to develop, can take up to a week to develop, but together they make sure that you can fight an infection. So all these cells and the whole immune system can be seen as a diffuse body-spanning organ, and it's quite a big organ because the mass of all immune cells in the body equals the mass of the brain. And this immune system can be divided into three types. We have solid tissues that store immune cells or where immune cells develop. We have floating tissues or cells. Those are the immune cells that are circulating. And we have fluid molecules that have specialized functions in the immune system, for instance, antibodies or the complement system that we'll talk about more in detail later on. So when we talk about lymphoid tissues, we have two types. We have the primary lymphoid tissues, and that's where immune cells are born and mature. And we have secondary lymphoid tissues, and that's where the mature immune cells are stored and waiting around for when they are called in action. So primary lymphoid tissues are the bone marrow, where all immune cells are born, and the thymus, where T cells specifically develop. And the secondary lymphoid tissues are either encapsulated, and then we're talking about the spleen and lymphoid, or unencapsulated, and those are the tissues that are in contact with the external environment a lot, like the gut, the lungs, the skin. Those have a diffuse immune system, which is called the mucosal associated lymphoid tissues, or MALT. And for the gut, this is specifically the gut associated lymphoid tissues. And those are present because these tissues are exposed to the outside world a lot. They have to fight a lot of threats, so they have a specialized immune system close by to help them out. Again, the difference between the primary lymphoid tissues and the secondary lymphoid tissues. The primary lymphoid tissues are the bone marrow and the thymus, and that's where immune cells develop and mature. And in case of B and T cells, also where they are educated. For instance, they learn what is self and what isn't self, so they can make a distinction between what is yourself and what isn't, and what has to be fought. The secondary lymphoid tissues is where the educated lymphocytes go, and where they meet antigen. And what is antigen? Antigen is a substance or any molecule that induces immune response in the body. And actually the word antigen is derived from antibody generator. So that's specifically for B cells making antibodies, but it's become a more general term for any molecule that induces an immune response. And an antigen is often foreign or toxic to the body, for example bacteria or viruses, which once in the body attract and is bound to a respective or specific antibody. But it can also be a cell, it doesn't have to be an antibody. So the primary lymphoid tissues, that's where lymphocytes and other immune cells are born and mature. For the bone marrow, all immune cells are generated there. So myeloid cells, lymphoid cells, like B and T cells. And the thymus is only for T cells. It's where the T cells mature and where they undergo education and selection for recognizing self and non-self. So it's easy to remember where B and T cells develop. B cells in the bone marrow, T cells in the thymus. So the lymphoid tissues that we have, the spleen and the lymph nodes, have different functions. So the lymph nodes are part of the lymph circulation, and the lymph circulation drains tissues and collects microbial antigens and cells and deliver these to the lymph nodes. So all the tissues in our body are flooded with veins that bring the blood, and the blood will give fluids to the tissues, but these fluids will also have to go away again, otherwise your tissues will be flooded and will grow. So the fluids are collected again in the lymph and drained to lymph nodes. And these fluids also collect whatever threat has started up in the tissue. And they also take along with them immune cells that can warn lymphocytes that there is something going on in the tissue. So immune cells, so these neighborhood watch immune cells in tissues, they can sense and intercept pathogens that they can then bring to the lymph nodes and tell the lymphocytes in the lymph nodes that there's something going on in the tissue and that they need help. So the lymph nodes are a collection of immune cells that are presented with antigens from the tissues. So what if an infection is directly going into the blood? Then how can you protect yourself against that? And that's what we have the spleen for. The spleen checks the blood for problems. So the spleen actually is one big lymph node. It's very similar to a lymph node, and it's highly vascularized, and everything that is in blood is going through the lymph node, through the spleen, and the spleen then checks if there are antigens that we need a response against, and also immune cells that carry threats are collected in the spleen, and they can then tell the lymphocytes in the spleen that they need help. Spleen also filters out rubbish like aged red blood cells. So in the lymph nodes and the spleen, there are B and T cells. Now the B cells can directly react to antigens. So if they recognize something that they think needs a response to, they can directly respond to that. So here's a B cell that can directly recognize an antigen, and if that is the case, the B cell becomes activated and starts producing antibodies against this antigen. The T cells, they cannot react directly to an antigen. They need somebody else, a dendritic cell, to present that antigen to them. So dendritic cells from tissues eat up antigens, then go to lymph nodes and present that to T cells, and then the T cells can decide whether they want to respond to that. And if the B and the T cells recognize something, then they start responding, and that is usually visible by these lymph nodes growing in size enormously, because when the B and the T cell become activated, one of the first things that they will do is start proliferating. They will start dividing to make sure that there are enough cells to help out with an immune response. And that can be seen here. Here we have the naive lymphocyte and the naive T cell. The naive B lymphocyte recognizes antigen and then goes through clonal expansion, which means that it will divide, so there are many of the same cells. The T lymphocyte doesn't recognize the antigen directly, but it's a dendritic cell that presents the antigen to the T cell. The T cell then becomes activated and starts going through clonal expansion. Then they will differentiate, so the B cell will become an antibody producing cell, and the T cell will become an effector T lymphocyte. They will start their fight, and they will eliminate the antigens or the infection. And when that is the case, then all these cells are no longer needed, and the majority of the cells will go into apoptosis, and a few of the cells will survive and become memory cells that will help when you're infected with a similar infection again. So these were spleens and lymph nodes. For the tissues connected to the outside world, these have their own lymphoid structures, like I told you before. In general, these are called mucosa-associated lymphoid tissues. They're very much like little lymph nodes inside the tissues, and they help with a faster response against threats. So in the lung, they're called bronchus-associated lymphoid tissues, or BALT, and in the gut, they're called gut-associated lymphoid tissues, or GALT. So all these cells and structures together make sure that you have a good immune response against an infection, and we'll talk about that later in the other recorded lectures.